So this was actually the title slide I gave to the Maine Water Conference. And the reason it's up there is to point out that the work I'm talking about is a collaboration with lots of people, right? There's a whole group of conservation groups that have been involved in our lake work, and they are range from very small, completely volunteer organizations to fairly sophisticated organizations with budgets of $500,000 a year to universities. And a lot of this work was funded under the EPSCoR, the previous EPSCoR grant to the state of Maine, which focused on sustainability science. And so the work that we're talking about is really about how we are going to sustain or how we're going to keep our lakes golden. And if you want to learn more about the work we're doing, and there was an encouragement to send additional resources, this is a nice piece that Maine Public Broadcasting did on our work. And it is a half-an-hour documentary on saving our lakes. So that might be a good place to go afterwards if you're interested in some of our work.
Whitney, can you do me a favor, just take off the glasses so that I click the...
Good. All right. So many of you have seen or seen the movie or read the book on Golden Pond. And Great Pond was the lake that the author was referencing in Golden Pond. So we're working on Golden Pond. So thus the play on keeping our lakes golden. And this is the iconic picture that people have of Maine lakes, these beautiful sunsets, the Walter, the fish, and Maine lakes, particularly the Belgrade Lakes, play a huge role in the economy of central Maine and the economy of Maine in general. This is, the chair sitting on the deck, the license plate, and this is the view looking south from one of the mountains on the north end of the lake. The economic impact of Maine lakes to the Maine economy is somewhere between $3 and $5 billion a year, depending on how you include the fishing revenue. So if you exclude the fishing revenue, it's probably $3 billion a year, and if you include the fishing revenue, it's probably $5 billion a year. It's comparable to the lobster fishery, or maybe twice the lobster fishery. So it's a big component of the Maine economy. And if many of you don't live in the state of Maine, but this would be a similar economic value if you were upstate New York or Vermont and other states with these classic lake systems. Certainly Wisconsin or Minnesota. The challenge is that our lakes are in peril. We have loved them to death. People come in, they build houses because the lakes are beautiful, and their living along the lake is adding to cultural eutrophication of the lakes. And you can see this now with Google Earth. So this is just a Google Earth image of North Pond, which is on the north end of the Belgrade chain, and East Pond, which is slightly east of North Pond. Okay, so we're not very creative in our names, but you can see the difference in the coloration of these two lakes on a Google Earth photo. And if the resolution, and if you're sitting close enough, you can actually see windrows of algae on this lake. And this is a yuck factor, which has significant economic impact. I mean, which lake would you buy property on? Historically, this was the preferred lake. The vista's better, the properties were nicer, but this is going to bloom. It's looking like this today, and will look like this for most of the summer. So this has a real impact on the economic value and people's perception of water quality. So the lakes I'm going to talk about are an interconnected chain, and that's kind of cool because it allows us to understand how the ecosystems are connected as we flow downstream. And this is the sort of schematic. We're going to be talking about lakes here, and this talk is really about lakes in general, but I'm going to frame it in the context of the Belgrades. And the Belgrades, this is East Pond and North Pond. This is Great Pond, and I'll show you some images later of the lakes in a slightly different projection. But the water flows kind of in a counterclockwise way through here, and these are what are called hypsographic profiles. It is the shape of the lake. So if you think of a lake as a coffee cup, it's the shape of the coffee cup. So East Pond and North Pond are kind of like shot glasses. Great Pond, which is what the movie on Golden Pond was based on, is kind of like a big, I guess, margarita glass. And we got a couple of champagne flutes in Salmon and McGrath and Snow Pond and Long Pond. But the important point here is that when you work in an interdisciplinary team as a biologist or as a chemist or as a geologist, you also need to incorporate the physics. And I think the other point I'm going to make is you need to incorporate the people, and Barry won't be surprised by that. And so it's the combination of all of these disciplines working together is really necessary to understand what's going on. So one of the first things we did is we made 500,000, I don't know where that data is, Susie, 500,000 measurements of depth of these lakes. So we went out with a boat and a GPS and a depth finder, and the students drove up and down the lake for a summer. Wasn't a tough summer job. And they measured the depth so we could contour the lakes, so we could generate these sort of profiles. They're called hypsographic profiles of the lake. The other part about data is that the state of Maine has a pretty good data set on lakes, and most states do. So that's sort of in the gray literature, but there's often really good GIS layers, there's often really good data layers if you know where to look. And so a lot of these profiles you can get at lower resolution through the state, in this case, the Lakes of Maine website. And they have data on about 1,000 lakes, and it's actively maintained by, actually, the Gulf of Maine Research Aquarium, I think, is where the server's located. So the important thing is that we've got some small lakes, we've got some big lakes. The bigger this profile, the more water that's in them. The number above them is the length of time the water spends in the lake. That has a big impact on the impact of human activity. If I put something into North Pond, 12 months later it's gone. But if I put extra runoff, phosphorus, into Great Pond, it takes almost 2.5 years for it to wash out. So you have a big thunderstorm event in August, which puts a lot into a lake. Depending on the size and what's called the resonance time, which is what these numbers represent, you get very different effects. So again, you got to understand the physics to understand the chemistry, to understand the biology, and that's important. And then the red line is the thermocline, and we'll talk more about what the thermocline is and why that's important. So the flow is sort of in this pattern going this way. The reason we care about the lakes is it's not just the flow of water, but it's the flow of dollars that also has an impact. And again, this lake is these lakes are worth a couple $100 million to the central economy, the economy of central Maine. In some of the towns around these lakes, 80% of the tax revenue comes from the lake. So if they lose 20% of the evaluation of the land around the lake, they're losing 20% of their tax base. And I don't think a lot of the residents understand that. There's a direct connection between the health of the lake and the economic vitality of these communities that live around them. Okay, so what's going on in the lake? I want to start with an analogy and make the point that life's controlled by the limiting reagent. So I'm a chemist, right? So life's controlled by the limiting reagent. How many bicycles can I make from 3 wheels and 2 bike frames? Audience? 1. I got 2 frames, but I ran out of wheels, right? Okay, so that's, all right, so see, that would be really happy if my Gen Chem students could just do that by the end of first semester. Okay, so there we go. So this is what a chemist thinks about. This is my limiting reagent reaction, right? So life, detritus, plankton are made up of 106 CO2s, 16 nitrates, 1 phosphorus, 120 waters, and that reaction is called photosynthesis. And photosynthesis produces oxygen, and it produces biomass. Okay, so the amount of biomass you produce is controlled by one of those limiting reagents. In a lake, water is not limiting. In a lake, carbon dioxide is seldom limiting. The 2 limiting reagents are either phosphorus and nitrogen, and in Maine lakes, the limiting reagent is phosphorus. That's not true everywhere in the world, but in Maine, it's a good assumption. Probably 95% of our lakes are controlled by phosphorus. And when you have too much phosphorus, this is what you get. This is a canoe paddle in East Pond. So that's what the satellite's looking at, and you can't see the bottom of the paddle. And that's a yuck factor. If you wade into the lake and you can't see your feet, you're probably not going to spend your next vacation on that lake, and you're probably not going to invest $300,000 on a piece of property, et cetera, et cetera. So this is the challenge, is that we need to regulate in an ecological way the nutrient budgets to restore these lakes that have tipped and to keep the lakes that are in decent condition from tipping into the future. So one of the classic ways people characterize lakes was using a technique called a Secchi disc. And this was invented by Giuseppe Secchi, who was the scientist to the Pope. And the Pope wanted to know what lake to build his private castle on. And they sent the scientist out to the lakes to evaluate them. And the story is his dinner plate fell into the lake, and as it was settling down to the lake, he watched it go and he watched it disappear. And he had an idea that I could determine how clear the lake was by attaching a dinner plate onto a string and moving them up and down in the water column until it disappeared. All right, so it's a pretty simple analytical device. Okay, and it actually works. You've got the sun, that's your light bulb. All right, the sun comes down, reflects off this disk, and your eyeball's the detector. And you measure how far down it goes into the lake, and the clearer the water, the deeper the Secchi depth. So this is the Secchi transparency, or the Secchi depth on East Pond, which is that green lake, for the last 40 years. So interestingly, the state of Maine has 40 years of data on these lakes, and I suspect many states do. So I was able to go into the historic database, and each point is a measurement made by a volunteer who's been well trained. And we start in 1975, and the Secchi transparency was about 5 meters, so that's really good. And you'll notice that there's a fairly narrow band. It goes from 4 to 5 meters, and then over time, that transparency has been getting less and less and less, but it's also been getting more and more and more. And so there's something interesting ecologically going on here. The trend is certainly up, but you'll notice I now have 7-meter Secchi transparencies when I never did, and I now have 2-meter transparencies when I never did. And this is because the lake is not a stable ecosystem anymore. It's oscillating between really clear and really green. So you might say that the lake goes from clear, interesting. This is the only slide. I think we're doing a sort of PowerPoint keynote transition here. So there was a picture of a green lake that was going to show up there. But what's happening is that we're getting this trumpeting of the Secchi transparencies. And as I start working with this data, I actually think that this trumpeting is more like this. It's a step function. And in 1988, something happened in the lake, and it transitioned from being sort of a classic Maine oligotrophic lake to something that is problematic from an ecosystem and community-based perspective. Other lakes in the Belgrades have more traditional Secchi transparencies, 7 meters, 5 to 7 meters, and these are not showing as much of a divergence. But in every lake we're looking at, almost in every lake in the state of Maine, we're seeing a 10 to 20% or more decline over the last 40 years. So the East Pond is sort of the cautionary tale, but all of our lakes are showing this decline. And this isn't just the volunteer getting older and needing glasses to measure the Secchi disc. But from a data standpoint, you worry about that, right? That's something the state actually worries about, is if you have somebody that's measuring this water quality for 40 years, I didn't wear these 20 years ago, right? But this data is real. So we're interested in understanding this dynamic, and how the plankton, in this case, this Gliotrichia colony, may be impacting that. Okay, so back to this question about where would you buy, right? This is this half-meter Secchi, 5-meter Secchi. The reason the picture looks black is you can actually see to the bottom of the lake. Okay, so this is now a little bit of Limnology 101, and this is designed to give you some context of what's going on in these systems. In a lake, in the winter, the water's cold in Maine, And as you enter the spring season, the whole lake is at 4 degrees C, because that's the density maximum of water. And so as you enter the summer, the bottom of the lake has cold, deep water, and it's at 4 degrees. And then as the sun starts to stay longer in the sky and the air temperature increases, mostly, however, it's driven by the sun, you end up warming the surface layer. And that warm surface water we call the epilimnon. We have a transition zone, which we're going to call the thermocline, and then we have the deep cold water, which we call the hypolimnon. And so we're going to have a transition zone in the lake from warm water, and you all know this if you've swam in a lake and you put your feet down and you feel the cold water down by your feet. What you're really feeling is a little bit of a seasonal, a little bit of a daily mixing. The warm water's at the top and your feet are down here. If you put the feet down here, it'd be like an ice box, right? I mean, this is 4 degrees, 42 Fahrenheit. You'd really know if you're down there. And the thermocline is that boundary in between. So let's give you an example of what typical Maine lakes look like. This is a series of different lakes in central Maine. This axis is depth in meters. This is temperature in degrees C, and notice that we're about 4 degrees. Even in September, in these really clear classic Maine trout lakes. This blue one is a classic Maine trout lake. It's called Clearwater Lake. And you can't buy property on that lake. Property is sold by word of mouth. It never gets listed. It's one of the Maine's classic, iconic brook trout fishing lakes, and the property gets passed through the families, and there's no outboards allowed on the lake. It's fly fishing, and so it's sort of this protected lake. It's up near Skowhegan. So that's Clearwater Lake. You can see the transition, that's the thermocline, and then you can see the warm layer. And so the warm layer in Clearwater is 27 degrees or so. And then the rest of these profiles are Belgrade lakes, and they all have that sort of classic profile. The depth of the profile is controlled by the size of the lake. The harder the wind blows, the more mixing you get at the surface. Year to year, that profile can move up and down, depending on how much sunlight you get versus how much rain versus how much wind. But it's all sort of in that range of 6 to 8 meters. It's also dependent on the amount of plankton that's in the lake. If there's a lot of plankton, they absorb all the sunlight right in the surface, and you can heat up that surface layer differentially. So if your Secchi transparency is only half a meter, the sun doesn't get down 8 meters, it gets down half a meter, and then it's all absorbed. If your lake is clear, your sunlight gets absorbed in the top 8 meters. So there's a biological component to this sort of map of temperature in a lake. So again, the geology, the physics, the chemistry, and the biology all work together to create these profiles of lakes. And this is all data that also comes out of the Maine Department of Environmental Protection database. And we've been adding to that actively this summer and the last few summers. Okay, so phosphorus is the limiting reagent. We have a thermocline, which defines a boundary between the top of the lake and the bottom of the lake. So here's my phosphorus. I got plenty of CO2. Photosynthesis, sorry, this is my biology slide. Bug. Okay, so I now did biology, and biology produces oxygen. And in a healthy lake, the phosphorus flows must balance, which means over any year, the inflow, which would come from a river or a stream, plus the non-point sources, that would be your lawn, the camp road. There are very few point sources left in Maine. A point source would be a dairy farm with manure running into the lake, or a outflow of a sewage treatment plant. Those have been pretty much dealt with. What still remains in our lakes is lots of non-point sources. The camp that was built by your grandparents and the outhouse was replaced with internal plumbing, and they needed a place to put the waste, so they dug a hole in the front yard and put a 55-gallon drum in there, put some gravel around it, covered it up with lawn, and it's never, ever backed up. And that's because that sewage is going straight into the lake, right? Because it's a nice gravel lake, and it never backs up, and if it never backs up, you never have to fix it, but yet it's still contributing phosphorus. That's the non-point source piece. And then the internal load, we're going to talk about in a second. That's phosphorus that's going to come from the lake. Now, those are the sources of phosphorus, which will allow us to produce bugs. And a healthy lake, that input has to be balanced by the outflow. And now you have a ecologically sustainable system. And if you have more outflow, you'd expect to have less bugs over time. And if you have less outflow, you'd expect to have a bloom over time. So that's the sort of paradigm. A small amount of phosphorus can produce a large amount of algae. Inflows need to balance outflows. If they're out of balance, then the rest of the lake comes into play. So we produce a lot of algae, and they're going to die over time, and they're going to end up in the deep part of the lake. And we don't have any oysters down here to eat them, and so these dead bugs build up. And as they build up in the cold water, they consume oxygen. So we have a mechanism to move phosphorus from the surface into the deep water, and that phosphorus builds up. And again, that's not terrible as long as we don't consume all of the oxygen, because all we're doing is moving this limited amount of phosphorus around a lake, and then in the fall the lake mixes and we reset the system. Where we get into trouble is we have now, we're out of balance, putting more phosphorus in the lake, we produce more bugs in the surface than the deep water can handle, and these organisms consume oxygen, and this reaction is called respiration, right? So we've got photosynthesis that's producing oxygen in the surface, and that oxygen leaves, goes to the atmosphere, because it's in gas, there's gas exchange, but there's no atmosphere down here. So the oxygen that's in this lake comes from mixing in the spring, and that amount is fixed. Okay, the oxygen in the deep water is all you got for the entire year. So if any of you have been to Moosehead Lake, which is this huge lake in northern Maine, they have a lot of oxygen in Moosehead Lake. The lake is deep, it's cold, and there's very little phosphorus. So the oxygen concentration in Moosehead stays high all year round. Sebago mostly stays high. And in our lakes, the oxygen's going away at a pretty alarming rate, and that has all sorts of other implications, because once the oxygen goes away, we can do a whole bunch of chemistry. So let's take a look at what the oxygen looks like. This is the temperature, and this is the oxygen profile. So I'm going to do the blobby thing here just for a second. This is the oxygen concentration in the surface of all the lakes, and it's all the same because they're all in equilibrium with the atmosphere. And this is what the oxygen should be in all of these lakes, because there's more oxygen in cold water than warm water. And this is what it is in Clearwater Lake. It's pretty close to the theoretical limit. So that's good news. And this is what it's like in all the Belgrades. Okay, so you come from this point, immediately at 8 meters it goes to 0, and then it goes all the way down. And basically, over time, we've produced enough phosphorus, or inputted enough phosphorus in these lakes, that we can basically suck out all the oxygen at the bottom of the lake. And once we do that, we can now start to consume the organisms that were not only coming down from the surface this year, but this muck, this organic rich material, is the accumulated insult to the lake over the last 100 years. So 100 years ago, these lakes were in balance. People moved in, they built camp roads, extra runoff, created extra productivity, and the lakes were still clear because that extra organic material just built up in the sediments and was removed from the system. But now, over time, we've gotten to the point where we now have taken out, we've got enough phosphorus, these bugs take out the oxygen, and once we remove the oxygen, the bottom water goes anoxic. And once the bottom goes anoxic, 169 references on that paper. That was on iron chemistry. And we have so this is a chemistry pun, this is the Ferris wheel, right? So once we go anoxic, the iron 3 goes to iron 2, and the iron 2 releases phosphorus that's bound up in the sediments. And I now have a mechanism to have a nonlinear biological response. I can drive these lakes into this unstable condition. And this is what's happening on East Pond, and it's what we're worried about happening in a lot of Maine lakes. It's not that they're going to bloom. this year. It's that we, over the last 50 years, we've insulted the lake and built up the organic material in the bottom that we have a high probability of them blooming over the next 10 to 20 years. And that probability is now high enough that there's a lot of concern about the sustainability of these lakes. So here's my paradigm. Go back to my cartoon: warm surface water, cold deep water. Phosphorus defines the phytoplankton. So I got a certain amount of phosphorus in the surface. And now the interesting thing is the phytoplankton help to define the thermocline. Because the more plankton I have, the higher up in the water column the thermocline develops. The thermocline helps define the deep oxygen, and everything below that thermocline now in our lakes are going anoxic. You can see that oxygen just went from saturation to 0, and it's in a zone about that big. As you scuba dive through it, you can see that. Dead bugs drive anoxia, and anoxia releases phosphorus through the Ferris wheel. So what have I done? I've taken the phosphorus that's been in this lake for 100 years, been input to the lake over the last 100 years, and I release it into the cold water. And it stays there all summer until the fall when it mixes. And now I put more phosphorus into the lake. And these lakes have residence times of 2.5 years. So all that phosphorus I release in the fall is waiting for me next spring. Okay, so phosphorus mixes upwards in the fall, and more phosphorus is available next year, and next year I get more phytoplankton. And that phytoplankton then has a physical impact on the thermocline, and this drives a nonlinear process. This cycle is highly nonlinear, and this is sort of what's happening in our central Maine lakes. Is we have this, and particularly in East Pond, we have this trend where everything was really good, and then all of a sudden there was a step function, and the lakes went from clear to really green in a period of a year or two. We think of ecological change as being gradual. That's seldom the case. There are certainly models where ecological change is gradual, but often the ecological change is quite dramatic. It's a step function, and we have a pretty good idea of the mechanism that can drive that step function for Maine lakes. Okay, so a way to characterize the sensitivity of the lake is this term called an Oxic Factor. And so what's happening is the ecologists and chemists and physicists working on lakes are trying to now develop extracted parameters that we can use to understand water clarity. And so these are computed values, and an Oxic Factor is the areal extent of the lake that is anoxic. So if half of the lake was anoxic, you would give that the lake an anoxic factor of 0.5. And if that was anoxic for 10 days, it would be 0.5 times 10, and that would give you an anoxic factor of 5. So if you have an anoxic factor of less than 10, that's good. That means that the lake only went anoxic for a short period of time, and if you have oxygen in the deep water, you can't release the phosphorus. If you can't release the phosphorus, you can't create a bloom. 20 is of concern, 30 is bad, 50 is awful. The reason I'm talking about this is that the story is we started to compute these anoxic factors for all the Belgrades. And once we did this last summer, we could show people where their lake was, or where their lake is relative to other lakes, and how the anoxic factor helped to predict the Secchi transparency. And that's what I'm going to show you. I'm going to show you a couple of slides that has a clear lake, Clearwater, has Ace Pond, which is sort of our cautionary tale, and then shows you some of the other Belgrades as potential problems. So what we're going to do is we're going to look down on the lake. This is a bathymetric map of Great Pond, and the red is where the water is anoxic. It's the deep part of the lake, and we're going to ask what percentage of this lake is anoxic, and on this day it was about 30%. So 30% of the lake bottom was covered by 0 oxygen water, so we would give it 0.3, and we integrate that over the whole season to come up with the anoxic factor. Okay, so Clearwater Lake, this classic trout lake, has an anoxic factor of 0. It never goes anoxic, ever, and it has a Secchi transparency. It's a little hard to see, but about 8. So that's great. You can see 8. That's 25 feet down in the water, right? So you can see the trout you're fishing for. China Lake. If any of you are familiar with Central Maine, China Lake is the water supply for the city of Waterville, and it's really gross. It's really green. It's in 1970, it had a Secchi transparency of 8 meters. And in 1983, notice this is Secchi, boom. This was not someone needing glasses. This Secchi transparency went from 8 meters down to 2 meters in about 6 years, and now it's stayed at about 2 meters ever since then. And the anoxic factor in this lake has gone from essentially 0 up to 80. And so this extracted value is doing a really good job at predicting this bloom. If we go now to some of our Belgrades, the Belgrade Lakes are starting to see this decline in Secchi transparency and an increase in anoxic factor, and it's right in the range that we predict will start to cause a transition. So this figure fundamentally changed the attitude about lakes for the audience, the homeowner audience around these lakes. So we were generating these plots last summer. I started to put these up, and all of a sudden people went, wait a minute. You know, this was China Lake, and when the anoxic factor got up to 40, it bloomed. And this is my lake, and the anoxic factor is up to 40, and I just spent $500,000 on my house, and what's going to happen? And it changed the whole conversation about water quality, and was it important, and how are people interacting with the lakes? So we went from, oh, it's very nice that you're studying our lakes, to a very different attitude. So I'll just skip. We've got some other anoxic factors. But it went from how do we study them to how do we fix it? Okay, and that's very interesting. As a scientist, we're pretty good at the studying part. It's when they turn around and say, okay, can you fix it? And there's an interesting story. We've been working on these lakes for a number of years, and I got a phone call one day from a woman who lived on East Pond, and she called up and says, hi, I heard you're working on the lakes. I said, yeah. She goes, good. And you're trying to figure out strategies to improve water quality? I said, yeah. She goes, good. My daughter's getting married in 2 weeks. Will you have it fixed in time? Right? And you kind of go, whoa, right? There's a disconnect here. Thank you very much for that compliment, but there's no amount of money that's going to fix that lake in 2 weeks, right? So it's interesting in dealing with the community. How do you communicate what you're doing in a way that engenders confidence and strategy and cooperation? This is really important. I'm used to writing chemistry papers, right? And this work has really forced me to get out of my comfort zone and look at how the community interprets our data and the type of things we're doing. So I'm going to talk a little bit now about how we're working on the communication piece. So the first thing is that the EPSCoR grant allowed us to put together a really great team of faculty. So we had faculty from environmental studies, biology, geology, chemistry. It allowed us to engage a lot of students over a 5-year period. We also had a lot of support from people like, this is Roy Bouchard from the Maine Department of Environmental Protection. Maine DEP has been beat up badly under the current political regime, but they're really a group of very dedicated scientists, and they're a great wealth of information. So that's if you're thinking about where the data is and what access is available to students and researchers, a lot of these federal or state agencies have a huge amount of data, and it's all in the gray literature. That's available, and they're a tremendous wealth of information. And Roy was a classic example of that. And Linda Bacon, who succeeded him at Maine DEP, has been equally helpful. So we brought in a whole bunch of tools to understand these lakes. And the important part is you're studying the lakes, you can't just be a chemist or a ecologist or a physicist. You need to sort of link all of these together. And so our project has incorporated education, facilities. We're fortunate to have the new Maine Lakes Resource Center, which is about a 4,000 square foot venue for communication and science. So we have science labs, but we also have venues to have the public come in. Free Wi-Fi and free public toilets. And it sits, I am standing on the boat dock. So you can come in from Great Pond and park your boat, go through this facility, and then there's a store on the other side where you can buy the New York Times and donuts and coffee. And so it's a place where you can connect the community. And as they pass through to use the Wi-Fi and the public toilets, that you can also have an educational opportunity. It's also a place where we have a farmer's market every Sunday. And so we can now pull people in and talk to them about what's going on in the lake. And let me talk about the trends. So that's been great. Collaboration across multiple institutions. Dissemination. We've been building a whole bunch of web-based tools that we hope people will use. And I'll talk a little bit more about those going forward. And then we've got all the normal physical measurements and biological measurements. And we're trying to integrate all of those together to try to solve this problem of declining water quality. So traditionally, you had a lake. Warm surface water, cold deep water, and you'd go out in a rowboat and you'd lower your Secchi disc, or you'd lower your sensor, or you'd lower your water sampling bottle. And anyone that's done field sampling, this is the sort of traditional paradigm. As we do with our students, we go out and collect water samples. And we bring them back to the lab and we analyze them. And that data has been invaluable at understanding the main lakes for the last 40 years. But that paradigm is changing. This is expensive. We like to sample in May. It's really cold. We like to sample in November. It's cold. If you look at the distribution of data, not only in lakes but globally, we tend to go to warm places to sample, and we tend not to go to cold, windy places to sample. So the globe is undersampled as well as lakes. So we need a new paradigm for sampling. The other challenge is that when we sample, we put it in a notebook and we put it in a database. It takes time to do that. So the state does that, and they actually sample Great Pond, lake we're interested in. And the last time that data is now available to the public, it was September 2012. Right? So that's 3 years ago. So there's a temporal disconnect between when we collect data and when it gets available. So it's in a good database, it's publicly available, but that database is 3 years out of date. So now if somebody says, what was the weather like yesterday? It was a beautiful day yesterday. You talk about the weather. We want to have that same conversation about lakes while people are on the lakes, while they're in a position to change their behavior. And you can't say, 3 years ago the lake was kind of bad, and motivate behavior, right? So we need to have a connection to people that is more current. And this is the database piece. And you want to own your data and you want to have control of your data, but at the same time you want to share it. And that's a whole nother discussion about who owns the data and how do you get it out there quickly enough that you change behavior, but not have your researchers that don't have tenure yet have their data scooped up. So here's one way we're doing it. Let's see if this works. I think it's going to work. Give it a second. This is a GIS map called Maps for Good. Maps for Good is a new company that is trying to connect people and place together. And this is a map of the Belgrade Lakes. It looks like the resolution may be a little hard to see, but if I cursor over to this flag, this tells me that on June 12th, these were the conditions in the lake. So that was the last time we sampled that site. And now everyone in the watershed can come along and say, all right, what's happening to North Pond? Well, North Pond is 68 degrees. It's got a Secchi transparency of 4 meters last week. Its thermocline was not well defined. It's a shallow lake. Its bottom temperature was 66 degrees. That's why there was no thermocline. The lake's uniform top to bottom. Its deep oxygen is at 96%. So now we've had that conversation. 96% oxygen is good news. We'll come back to Great Pond. Its deep oxygen is at 71%, and as of yesterday, it was at 52%. So the idea is we're trying to create these tools which people can use that are current, and they can compare lakes to lake to lake, and they can start to see the trend, and now they can say, whoa, 71%. Well, that tells every fisherman that there are no salmon and trout in this lake. Browns, maybe, because they'll sit above the thermocline, but all the cold water fish have to hold their breath for 3 months. And so the cold water fishery in these lakes is the traditional salmon fishery in these lakes is significantly impacted. So this is new. We just sort of put this online in the last week. We also do things like put hiking trails on, because the hiking trail, click on that, it tells you about the trail, and it will tell you the map, because people come to these information sources for lots of reasons, and if I can convince them that hiking is interesting, then they can also go and look at lakes or vice versa. So there are lots of opportunities, and so this is, again, a small company, and they gave us Dropbox. We use Dropbox to update the data. We just change a file in Dropbox, and 5 minutes later, it's up and live on this page. Okay, well, now I need my glasses. I need to go back to lower left. Okay, right there, keynote, right? Good. So that's one way we're trying to communicate. The other way we're generating data on a sort of daily basis is with buoys. And Barry, you mentioned buoys. And ours are actually within cell phone range, so that makes things easier for us. So there is a buoy floating out in the middle of the lake, and guess what? It's called Goldie. It's on Golden Pond. So one of my colleagues likes to name everything. So the buoy has a name. And the buoy has a whole bunch of traditional water quality sensors on it. And so there's a whole area of engineering and chemistry. There's actually some really cool biological sensors now that are coming out in the literature. And these are designed to give us real-time pictures of what's going on. So that's something to also sort of keep in mind, is that our traditional way of sampling is changing as technologies have improved. And there's a cell phone sitting inside this buoy that's sending me data every 15 minutes. And so now we can create displays. And one of the challenges we're going to have is to take this, which is the display that comes from the buoy. The engineers that built the buoy love this display. This is not particularly impactful to a diverse audience, okay? And I really like this display. This is the temperature profile in the lake over the entire summer. So this is the bottom of the water, and guess what? The bottom water stays cold. And this is the surface water. It got warm, and I can look at this and say, wow, there's a thermocline in here, and look at the daily variations. This is not useful to a general audience. So we're really working on ways to interconnect all of these various information streams. So we will take our buoy, we'll take the raw data, we'll send it to one of our servers. We use National Instruments LabVIEW, so we've got some computer science components in this project to integrate it, to create images that are maybe, hopefully, more useful to a general audience, and then we can actually create cartoons to show people what's going on. And so the best example of that is actually the Weather Underground, right? So the Weather Underground is a great example of a display of complex data in a way that resonates with people. And this is my attempt at creating a Belgrade Lakes cartoon. So the red is the warm surface layer, the green is the thermocline, the blue is the deep water. I've put the oxygen concentrations on here, and this windsock changes directions as the wind changes, and my fish goes from a happy salmon to a sad fish when the oxygen drops below 70%. The nice thing is that we can change this. We can test it. We can run focus groups and say, do you understand what this diagram shows? The maps for good that I showed you, we did that last night. 40% of the audience understood it. 60% did not. We're like, whoa, what do you mean? You know, these are people that have been living on these lakes their entire lives. I've given, I don't know how many presentations at their lake associations about Secchi transparency. They're like, what's Secchi transparency? Right? So there's a real challenge in communicating to a general audience about what's going on. And so there's a real need to create fact sheets and handouts and links to expand on the data. So I look at my cool cartoon and I go, look at all that information, and I'm not convinced it's going to communicate what I hope that I'm communicating. So there's a good lesson in that. So this is sort of a process profile, and I hope this communicates sort of what's happening in the lake. This is time, this is depth, and this is heat map. And the brighter the color, the hotter the water. And this is what happened in 2014 in Great Pond. So in the spring it was mixed. This is the thermocline. And then in the fall it cools down and it mixes again. And there's a hot zone right there, and this was 2013. So you can now compare 2014 and 2013. You could say, ooh, it was warmer for a couple of weeks in 2013. And we can generate these dynamically. And now the question is, do people understand that? And we can put oxygen maps on as well as temperature maps. So we can do all kinds of things. So this is this is the gold standard. I'm going to say Weather Underground's the gold standard. I don't think we're there, but that's an interesting area of research. It's not just enough to generate data anymore, or even to have a really good database to put it in. It's really important in how you communicate it, particularly if your audience is the Belgrades, and they want their lakes fixed. There's a lot more we can do with these buoys beyond just collecting data. The high-resolution data allows us to learn more about the lakes. And I'm going to skip a bunch of slides because looking at the clock, I want to give some time for questions. This is just so you know where I was going. This red line is the oxygen data at the surface, and this blue line is the oxygen data in the deep water. Right, so you can see this anoxia happening, and it's down to about 20% here, and it goes completely to zero until the lake mixes, and then it goes shooting right back up. But there's all sorts of interesting research questions that you can do with this data, because notice how it's going up and down? This is the lake inhaling and exhaling every day. Photosynthesis produces oxygen, respiration consumes oxygen, so you can do all kinds of cool things. Like if you zoom in there, this is it going up and down. Photosynthesis, respiration. And if you know how much oxygen's going in, you know how much plankton's being produced. And if you know how much plankton's being produced, you can calculate what's called gross productivity and net productivity, and you can tease those numbers out. So you can learn these buoys not only give you freedom in terms of not having to be on the lake every day, but they give you that high-resolution time data set, temporal data set, that you can extract a whole bunch of additional physical factors in. So, and I'm going to skip this. This is all the calculations. So if we want to know more about gross and net primary productivity, we'll take you through this during the questions. Now you know why I skipped it. Okay. So the work we've been doing over the last 2 years, particularly that one slide on anoxic factor, fundamentally changed the community's interest in the lake. And I presented that in October, and by January, the community, and this is in the inside of the Maine Lakes Resource Center, had hired Dr. Ken Wagner. He's an international expert on limnology and on lake restoration. That's what he does. He comes in and says, he's a paid consultant to fix lakes. And the community went from, it's very nice you guys are studying it, to, I want you to fix it. And so he came in and he looked at the data, said, yep, you got a problem, and we can fix it. And it's going to cost you $10 million. And I went, whoa. And half the room went, whoa. And the other half of the room went, that's all? They went, that's only $500,000 on the tax base. And that was interesting, right? And so we went very much from our lakes are fine, stop telling us that there's a problem, to, whoa, I want to fix it. I want to fix it now. They wanted a plan to go in this summer to fix the lakes. And that was interesting. That was a social change that just was like a line in the sand, and it was one slide. So the communication piece is really important. Once they got this idea that anoxia was a real problem in the bottom of the lake, and they started comparing it to lakes that they had experienced as having nasty algal blooms, they wanted it fixed. So how do you fix it? Well, the first thing you do is you model good behavior around the lake. So this is the Maine Lakes Resource Center, looking at it from the lake, and we have, these are native wildflowers, mostly native. And this is a buffer to keep the nutrients from running into the lake. This building was built with porous pavers that infiltrate water to keep them from running into the lake. It was built with a full, fancy anaerobic digestion, sort of active septic tank that could deal with the extra nutrients from the lake. So one of the ways you do it is model good behavior. And one of the concerns is that even if we're on our best behavior from now on to the end of forever, the lakes have already gotten to the point where they're not recoverable. And so that's one of our goals this summer, is to understand that. So if they're not recoverable, what do? Well, one of the things you can do is inactivate the phosphorus. And the problem is anoxia, and iron gets reduced, it releases the phosphorus. But you can also bind phosphorus with aluminum. And aluminum doesn't have any redox chemistry. It's aluminum plus 3. And so you can actually add aluminum to the lake, it binds up all the phosphorus, problem solved. It's interesting. It's an engineering solution. For Great Pond, I think it'll probably cost $3 to $4 million. It'll last 25 years. And probably last 25 years. It doesn't come with a warranty. But that's something that they're actually thinking about. And so you can inactivate the phosphorus, because if it can't come out of the sediments, it can't create the bloom. It'll take 3 or 4 years for the residence time to catch up. But in many lakes, they've done this, and it's a pretty reasonable solution. The other one is oxygenation. This is the one that the consultant really is promoting. You actually run 20 kilometers of pipe in the lake, and then you put pure oxygen in. And if the problem is anoxia, you just add oxygen. It's $150,000 to $200,000 a year for oxygen. Seems like a lot of money to me, but not on a community-wide basis. The metaphor of a lake with a respirator comes to mind, right? And so that's where the community piece is going to come in, because the oxygen probably is going to be faster. You can turn it on and off. But once they start the oxygen, they're committed. They have to run it forever. They turn the oxygen off, the lake goes right back to where it was. Whereas the aluminum allows them to sort of tune it for 20 years. So this is interesting. In a year, we've gone from, the lakes are okay, to, wow, we want to fix it, and fairly dramatic engineering solutions to this in these iconic Maine lakes, which is really interesting. So that's the oxygen control. We just bubble oxygen in. You get a pipe, you put oxygen, you form little teeny bubbles. The bubbles never make it to the surface, but they add oxygen to the deep water, and the problem's fixed. And then aluminum, they actually go out with looks like a huge plant, like a fertilizer, except it's bags of aluminum. And they go up and down the lake, just like you would be spreading fertilizer on your lawn. And they add the aluminum, and the aluminum takes care of the problem. So that's interesting. So what are we doing this summer? This summer, as a consequence of all this, we have a whole bunch of students that are going out every week. They are our essentially student-based buoys. So we only have money for one buoy on one lake, so we're working on all the lakes, and we have students visiting every single lake once a week, and then we're putting that data up on the website. We're working on a whole bunch of educational components, and then we're actually working on a comprehensive water quality plan, which is due in February that's not a typo, 2016. And it's going to engage the community in where do they want to take their lake. Do they want to hold the ground, just keep it where it is? They want to take it back to 1980? They want to take it back to 1950? How much money do they want to spend? Do you want to create a water conservation district in central Maine? We've got 7 towns around these lakes. Think about if any of you from Maine, there's a whole business about the budget. And it's going to be a really interesting political football in terms of we want to increase your taxes so that we can increase your property values, and the community and all the people that live in the towns, not just the waterfront people, but the economy of that central Maine is based on the lake, the people that put in the docks, the people that lead the fishing trips, the people that do all the maintenance. Again, those folks, all those support jobs are all based on this. And we've been fortunate that, oh, I can say this at Bowdoin. Tom Klingenstein is not a friend of Bowdoin, but he is a friend of Colby, and he is funding all of this work because he has a house on Great Pond. You can look up Tom Klingenstein in Bowdoin, you'll understand the... yeah! Yeah, he's the golf guy. Yeah, okay. So there's an interesting story on that one. We're making good use of it. Okay, and so that we're trying to take this resource center and this web and create a land buoy. So I think that's it. So I'm done.